As most of you must know, Dr. Kanneganti is a world-renowned researcher in the area of innate immunity, especially inflammasomes. So thank you very much for this opportunity, Dr. Thiru, and we're happy to hear you.
Thank you. So I thank again to Diego, Gustavo, and all the members there for the opportunity to present our work today. So I'm going to discuss innate immunity, our first line of defense against a wide range of pathogens. And it's not only about innate immune recognition, but also how innate immunity triggers inflammatory cell death, the mechanisms involved, and their role in disease. So before I really start presenting, first I would like to thank all my former group members as well as current members for their contributions. So 30 of my group members have become faculty and group leaders at major universities and pharma around the world. And the topic I'm going to talk about is because of their hard work and also our current lab members who are contributing towards the concepts I'm going to discuss. So to just give you an overview of innate immunity, which is our first line of defense, what we know is we encounter pathogens, or you all know about pathogen-associated molecular patterns or DAMPs. And once we sense any of these danger signals, then our innate immune sensors sense the danger and form these multi-protein complexes that are essential for driving cell death, which is lytic and inflammatory. And it's inflammatory because it leads to the maturation or a release of several of the cytokines, inflammatory cytokines, mounting an immune response. So is this as simple and straightforward as you see here? Of course, it's not, because there are, what I think about infections as limitless number of triggers, right? So there are bacteria, there are viruses, they have parasites, fungi, and they all have very different lifestyles, and the way they infect cells, the way they hijack host immune system or host cells is really fascinating and very unique to each of these pathogens. And also the way they enter the cells, that could lead to damaging cells or lysing cells that will further lead to release of pathogen-associated molecular patterns or DAMPs. And endogenously also, there could be accumulation of danger signals, such as, for example, ADAR1, our editing enzyme, doesn't work properly, then there is accumulation of nucleic acid, which is a danger. And there's accumulation of any of metabolites is a danger. So our immune system is tasked with sorting out all of these triggers, which we can say that limitless number of triggers, how our immune system sorts these out and how it decides to either, lead to cell death or not. It's a fascinating research area, and that's what I'm going to touch upon. But then before even we go into innate immune sensor or innate immune cell death, mutations at every step of innate immunity and cell death leads to disease, right? So for example, mutations in innate immune sensor molecules like NLRP3 or NLRC4 lead to autoinflammatory diseases. And also the signaling pathway molecules, mutations in signaling pathway molecules like caspases or ribcase are also involved in driving inflammatory responses. And the cytokine pathways that are downstream of these innate immune sensors are downstream of these signaling pathways also lead to several of the autoinflammatory diseases. And then not only the mutations, any alterations to these pathways would lead to infectious diseases, several of the autoinflammatory diseases, neuroinflammation, and also cancer. So you can see and appreciate how important it is to understand innate immunity and the pathways that are driven by innate immune sensors. So for the last 3 decades almost, we've been trying to ask two fundamental questions. One is like, how do we sense this limitless number of triggers? So how our innate immune system can sort out this limitless number of triggers and how cells die. And these two important questions have implications towards the way we understand maintenance of homeostasis or when the responses are altered in such a way, it could also lead to inflammation, morbidity, and mortality, right? So these two important central questions that we've been addressing in the lab. And then there are also several of the sensors that we could focus on. You all might know about the toll-like receptors that are way upstream on the cell surface. But my lab focuses on studying the cytosolic innate immune sensor family members called NLRs. So as a PhD student, I studied plant NLRs. And then as soon as mammalian NLRs have been discovered, I switched fields from studying plant NLRs to that of mammalian NLRs. And as you can see here, plant as well as mammalian NLRs, they share a lot of features. As you can see here, the central NAC domain is there for oligomerization, and then C-terminal leucine-rich repeats. And the fascinating aspect of these sensors molecules is that they have N-terminal card, pyrin, or bit domains that are essential for protein-protein, like interactions and forming complexes. So the N-terminal region dictates the type of complexes that can form or bind to induce inflammatory responses and cell death. So when we entered the field, it was wide open as nothing was known at the time except for a couple of molecules. And I was really interested in understanding role of NLRP3 because at that time it was an enigmatic molecule. Not much was known except that there are mutations in these patients where NLRP3 mutations lead to several of the periodic fever syndromes. And we always believe that diseases provide a foundation to discover mechanisms. So we started with NLRP3 to really understand what, in the broader context, NLRs do and their role in driving these autoinflammatory diseases. So in our first study, we, together with Dr. Vishwa Dikshit's lab and late Dr. Yerk Chop's group, showed that NLRP3, this study provided the first genetic evidence for the role of NLRP3 in activating caspase-1 in response to microbial components. So of course, we know that there are these patients, but these studies showed that there could be triggers, specific triggers, activating NLRP3 to form this multi-protein complex called inflammasome for the maturation of pro-inflammatory cytokines IL-1 beta and IL-18. And the inflammasome term was given by the late Dr. Yerk Chop, and he defined inflammasome as a multi-protein platform that triggers activation of inflammatory caspases. This is a definition of inflammasome and also pyroptosis that I'm going to discuss later on. So a molecular platform triggering the activation of inflammatory caspases. So here, of course, caspase-1 and processing of IL-1 beta and IL-18. So at the same time, even though these three papers published back to back, the triggers that all three papers showed were very different. In Dr. Yerk Chop's group, they showed MSU crystals can activate this pathway. And then Dr. Vishwa Dikshit's group showed LPS-ATP, and we showed imiquimod and microbial components can activate NLRP3. So it didn't make much sense at the time, like how NLRP3, one of the sensor molecule, which should act as a receptor, is sensing these wide range of microbial components. So we asked a very simple question: is there any physiological relevance to these microbial components or the triggers that we are seeing? And can live pathogens activate NLRP3 inflammasome? So why live pathogens? Because we know that LPS is not equal to pathogen. So if you look at a pathogen here, which is Salmonella, you can see the PAMS, pathogen-associated molecular patterns that are there, right? Like there is flagellin, it can release toxins. Salmonella can bore pores using type 3 secretion system. It's a gram-negative bacteria, so it has LPS. So, and then when bacteria are getting degraded in the host, many of these PAMs are released. So trying to just use LPS and to say that we are studying innate immunity really doesn't make much sense, because you are completely ignoring all the rest of the factors. You can see if you believe LPS is a pathogen, it's like looking at the tail and thinking it's a rope and ignoring this big elephant in the room, right? So we don't want to be really blind men and the elephant. So we wanted to really ask the important question about what are the pathogens that can engage NLRP3 inflammasome. So we screened at the time many pathogens that we could test, and of all the pathogens, our data suggested that influenza virus can specifically activate caspase-1 and induce maturation of IL-1 beta, IL-18 via NLRP3. So this is the first study to establish that a live pathogen can act as NLRP3 inflammasome trigger. This goes way, way back to 2 decades almost. Then we started asking the question about the role of NLRP3 in other diseases because the patients have, they develop autoinflammatory diseases. So we started establishing all these in vivo disease models, like where Jackie and MRK were interested in asking the question about the role of NLRP3 in infectious diseases like Candida or Aspergillus, or in cancer model, especially in colon inflammation. Rinki came from Wageningen University. He was interested in understanding the role of NLRP3 in metabolic diseases. John was, really intrigued about understanding the role of NLRP3 in adaptive immune responses, autoimmune or other autoinflammatory diseases, and joint inflammation. So all in all, at that time, we established maybe around 50 to 100 different models to ask the specific function of NLRP3. And I think these studies and many others establish the important role of NLRP3. And I think many of these studies on NLRP3 really helped to push the inflammasome field forward, because these studies told us the importance of NLRs, or especially NLRP3, in health and disease. You might ask, is this reflected in therapeutic pipeline? And of course, every single company here, your favorite, you can see it there. They all are interested in identifying inhibitors of this pathway. So going back to our patients, and as I mentioned, we always look at diseases as a way to figure out mechanisms. And there are many bone diseases. Of course, mutations in LRP3 leads to bone disease in these patients, but also there are many other bone diseases with no targeted therapeutics. So John asked a very simple question about the role of the cytokine dedicated towards inflammasome activation, right? The dedicated pathway for IL-1 beta maturation. So John asked whether IL-1 beta has anything to do with this bone disease. And to ask that question, he developed a mouse model, which is chronic osteomyelitis bone destruction model, where mice, when they're born, they're normal. But as they age, they develop this severe inflammation in joints. And when he crossed this PST-PIP2 or Chrome mouse model with IL-1 beta deficient mice, he thought like it will be protected, because we believe that inflammasome mediated IL-1 beta is the driver of this disease. And indeed, John was able to show that by removing IL-1 beta in this background, there's complete protection from bone destruction. So next we asked a very simple question, which is the role of NLRP3 inflammasome upstream. So in order to ask the question, we generated the mouse model where Chromo model is crossed with NLRP3 knockout mice, CASP is one knockout mice, and ASC knockout mice. So with our IL-1 beta knockout mice, it would be very clear that the molecules upstream of IL-1 beta would be required. So let's see the data. Here, it's a big surprise to us, which is completely against our hypothesis, or against the dogma in the field, that IL-1 beta cannot be matured without inflammasome, without caspase-1. And what our data is suggesting is that caspase-1 has nothing to do with IL-1 beta maturation or NLRP3 ASC. The complete NLRP3 inflammasome is not involved in driving this disease, because deletion of these molecules did not protect. So this was completely shocking to us because the downstream molecule is there. Then we went into rabbit hole thinking that, there are other proteases like cathepsins, calpains, protease 3, elastase, all that are expressed in neutrophils or macrophages could be driving this disease. But the long story short, we're able to see that after generating almost 30 different crosses, we're able to see that only these particular crosses were able to provide protection upstream of IL-1 beta. So these are extremely important findings because this laid the foundation for the concepts that I'm going to talk about, that opened up a wide research area in innate immune cell death field. So what we are seeing here is that NLRP3 and caspase-1 inflammasome molecules that we thought that are not involved are involved in a redundant manner with that of caspase-8 and RIPK3. So what's happening here is when we don't have caspase-1, caspase-8 is regulating IL-1 beta responses. And when we don't have caspase-8, of course, caspase-1 and NLRP3 inflammasome is regulating this. So this showed again the beauty of redundancies in vivo, and we would never be able to discover this phenomenon unless we used in vivo mouse models or in vivo models of disease, physiologically relevant models, right? So this study told us many, many things. And of course, we went back to our patients again, and they also developed skin disease. So we asked whether there is any role for inflammasome in skin disease. And our bone disease model told us that IL-1 beta could be the culprit. So we asked, first we developed a mouse model of inflammatory skin disease, which is PTPN6 spin model, where these mice, when they're born, they're normal. But as they age, they develop this severe inflammation. And even if you microabrade gently, this foot pad would become totally like pyoderma gangrenosum, phenotype. So we asked whether IL-1 beta is involved. And of course, a surprise that it's not IL-1 beta that is driving this disease. Then we asked if it's other IL-1, which is IL-1 alpha. Even after 6 decades of research, not much is known about this other IL-1 alpha. And our studies clearly showed that deletion of IL-1 alpha fully protects this PTPN6 spin mice from developing this inflammatory disease, skin disease like pyoderma gangrenosum. So this again, gave us a lot of indications about how inflammation works in vivo. And we started asking whether inflammasome is playing a role upstream of IL-1 alpha. And then again, against the dogma, it's not inflammasome, it's not NLRP3 or caspase-1 or ASK that are regulating IL-1 alpha responses. So this drove us completely crazy. You can see that in front of your eyes already there are 35 knockout strains. We in total maybe developed 100 different mouse models to nail it down and to nail down the pathway. So we generated the single, double, triple, quadruplicate knockouts, and we did discover the role of several important molecules upstream of IL-1 alpha in driving this inflammatory skin disease. So for example, you can see here that TAC1 deletion provided protection, RIPK1 deletion provided protection together with that. And then we also know that it's coming from MyD88 pathway, and the QKO that's fully protected is RIPK1, caspase-8, RIPK3. So all in all, the bottom line is, if you remember the bone disease, we have seen caspase-1, caspase-8, RIPK3 deletion provided full protection in bone, but also in skin disease. And more importantly, we're able to see that RIPK1, the key molecule in the complex, is also involved in driving this inflammatory disease. And if you delete RIPK1, there is absolutely no disease. So somehow this TKO is really driving, upstream of IL-1 alpha in driving this disease. So this work was done by John and Prejwal. It required a lot of persistence, I guess, to really reach to the stage of discovering key molecules in this pathway like TAC1, RIPK1. Again, laid the foundation for our future work on panaptosis. So at that time, I got a call from dermatologist, Dr. Lisa Beck, and a patient came in with this like they thought he got infected, but then they couldn't find any infectious agent contributing to this. While they were testing all different conditions or infectious agents and all of that, and cleared that there is no role for infectious disease, this person almost lost his finger and very close to amputation. But then as soon as we saw our studies on this skin inflammatory disease, they put this person on IL-1 blockade, and you can see that, the wound got healed and he was able to rescue his hand. So that's importance of really understanding. It's worthy of all our efforts to really understand the pathway and the molecules involved. And then switching back to NLRP3, by then we know that NLRP3 can sense a lot of anything that it can see. That's the joke in the field, that NLRP3 is central to sensing everything and anything. So that told us that NLRP3 is not working as a receptor, but it's acting as a sensor downstream of a receptor or another innate immune sensor. So we also showed influenza virus is triggering this specifically. And we also know that LPS-ATP or LPS-nigericin are specific triggers. So Prejwal started screening all those hundreds of different molecules or knockouts that we have in the lab to identify the key molecule that is upstream in this pathway. So again, against the dogma, a very, very big surprise that he found that caspase-8 is playing a central role in transcriptional and translational regulation of NLRP3 inflammasome. So this was totally against the dogma. This was back in 2014. None of the leaders in the field believed that there is a role for apoptotic caspase in driving inflammatory responses, and especially linking an apoptotic caspase to that of inflammasomes was completely against the dogma, against the belief of anybody else. But data is data, right? So our data completely showed a critical role for caspase-8 in this complex. So this study that was published back in days showed for the first time provided genetic evidence for the role of caspase-8 in caspase-1 inflammasome activation. This also told us many things, that there is plasticity and redundancy between cell death pathways, and also innate immune molecules can interact with other caspases or multipurpose molecules. So then, the key, I think the key factors here are, we always think that caspase-8 activation means apoptosis. So our data told us that it's not. So caspase-8 activation in lytic condition, if cell is dying in a lytic way, it is not apoptosis, because apoptosis is a non-lytic, non-inflammatory pathway. And always people get confused about, as soon as you say caspase-8, they link it to necroptosis. And the important fact here is, if caspase-8 is involved, it cannot be necroptosis, because necroptosis happen in the absence of caspase-8. So these are all misconceptions and confusion in the field, that they link caspase-8 always to either apoptosis or necroptosis. So it's not apoptosis or necroptosis if there is a lytic cell death pathway initiated or instigated by caspase-8 in innate immune complexes. So, and then also there's very clear distinction in the field that inflammatory caspases and then apoptotic caspases, they're clearly categorized. Caspases, which are central to cell death, are categorized into inflammatory caspases, such as caspase-1, 11, 4, and 5. And then, of course, you all know about initiators, caspase-8, caspase-9, and the executioners, 3 and 7. So this is the dogma in the field. And if you ask any cell death expert in the field, this is what they say: we have apoptotic caspases, inflammatory caspases, right? But then what is our data telling us? That there is no clear distinction at all, and we have to be open-minded in understanding and appreciating. So if you go back to the inflammatory cell death pathways, we know that innate immune cell death pathway that's established at that time is pyroptosis. So pyroptosis is mediated by inflammasomes, and Dr. Yerkchop's definition of inflammasome are, and then Cookson et al. Define pyroptosis as a pathway mediated by inflammatory caspase, which is caspase-1. So inflammatory caspase-1 mediated, gasdermin mediated death is pyroptosis. And then in contrast to pyroptosis, apoptosis is a non-lytic, non-inflammatory death. where caspase 8, caspase 9 drive 3 and 7 activation. And in contrast to these established pathways, necroptosis is a backup mechanism where when caspase 8 is inhibited, this happens only when caspase 8 is inhibited upon engagement of like TNF, then RIPK1, RIPK3 via MLKL drive necroptosis. So if a pathway is not dependent on MLKL, it's not like innate immune pathway. If a pathway is totally dependent on MLKL, of course, it's necroptosis, right? So to keep it straight, absence of caspase 8 leads to necroptosis, and we have inflammatory caspases associated with pyroptosis, and then this is non-lytic, non-inflammatory apoptotic pathway. So you can see there are very clear distinguished features for all these pathways. But then what our studies for a decade, almost for a decade, showed that or provided genetic evidence for the role of caspase 1 in cleaving caspase 7. Our data also showed caspase 1 can cleave PARP1. And as I just mentioned, caspase 8 could be acting upstream of this inflammasome, regulating caspase 1 and IL-1 beta. And then we also showed critical role for NLRP3 caspase 8 TRPK3 complex, or we can replace NLRP3 with caspase 1, caspase 8 TRPK3. So these crosses provided substantial evidence, or these are pressure tested through combination of genetics and biochemistry, that there is this intense crosstalk that is happening between caspases, and innate immune pathway utilizes all these molecules in driving cell death. So again, we were breaking all the rules set by the leaders in the field or experts in the field, and it's against the dogma to really talk about something that apoptotic caspases are involved in driving inflammatory cell death pathway. But data is data, so we went ahead to ask other important questions. But the problem is none of these established models could explain this crosstalk. So necroptosis doesn't explain because caspase 8 is involved in our pathway. Inflammasome pyroptosis doesn't explain because more than inflammatory caspases are involved in our pathway. So what do? I think we have to essentially go with the right model. So Dr. George Box, a famous statistician, says that essentially all models are wrong, but some are useful. So we always have to ask, is the model good enough for this particular application or not? And then we cannot just get married to dogmas in the field or established phenomenon unless we can explain. And to explain that, we always need to commune with our data. Just go with the data. So that's all we did. I mean, our own Peter Doherty always says that you start with your results, you look at your results, you live with them, you talk to your data, you commune with your data. So that's what we did. And also, we all like very simple models, right? And a innate immune sensor binding to ligand and driving one pathway makes sense to us. So if we complicate anything, nobody appreciates anymore. But whoever doesn't appreciate, they have to think in depth about this network. So you can just imagine how conceptually we miss a lot of things thinking that this is one tree or a trunk. But then here you can see the network or molecules that could be connected, that could be leading to the phenomenon in health and disease. So unless we really nail down the signaling pathway or the network of molecules that are interacting, we can never find targeted therapeutics. And that has been a major problem in the field of cell death and innate immunity, that so far we couldn't figure out the exact molecular combinations because we always want one molecule or one simple pathway. But life is not simple. It's all, like this network, right? So we went back to ask the question, how do we explain this complex? So in order to do that, we went back to our model, to NLRP3, and started asking very simple question. Is there an innate immune sensor that can engage molecules like caspase 8 and RIPK3 and also inflammasome to drive a cell death pathway? So in other words, we are asking whether influenza virus can induce lytic cell death via this complex. And if so, what is the sensor that is directly, or a receptor that can directly bind to influenza infection mediated PAMP release? So that's CBP1. So before we discovered ZBP1, it was a completely enigmatic molecule. No one knew exactly what ZBP1 does. And in our studies, we were screening with our influenza virus. And if you remember, we showed back in days, 3 decades ago, almost in 2006, 2 decades ago, that NLRP3 is involved in caspase-1 activation and IL-1 beta, IL-18 regulation. And our discovery of ZBP1 led to understanding of the role of ZBP1 in also activating inflammasome, which is caspase-1 activation and IL-1 beta maturation. So this could be an inflammasome, but we couldn't understand why when an NLRP3 inflammasome is there, what could be ZBP1 doing in addition to NLRP3 inflammasome? And that told us that ZBP1 primary role is to engage inflammasome for IL-1 beta maturation, but beyond that, it's involved in cell death. So here what we are saying is that influenza virus is inducing this lytic cell death that is totally dependent on ZBP1. And this is not pyroptosis because NLRP3, even though it's involved in caspase-1 activation for IL-1 beta maturation, it's not involved in driving cell death. So for that matter, none of the pyroptotic molecules like inflammatory caspase-1, caspase-11, or the executioners like gasomin D or E are involved in disease. So even when we delete, cell death was still happening. And obviously, this is lytic cell death. And we asked if there is any role for apoptotic caspases, and we found caspase 7, caspase 3 has no role in driving this lytic cell death, obviously. And then we also asked if RIPK3 and MLKL are involved. And our data showed, and other data also, that MLKL, deletion of MLKL does not provide protection. That means it is not necroptosis. And RIPK3 provides partial protection, and if you use increased dose or in a time-dependent fashion, even RIPK3 deficiency leads to this inflammatory cell death. So we found a sensor, and this sensor is activating NLRP3 inflammasome for IL-1 beta IL-18 maturation, and also driving cell death, which is not pyroptosis because it's not dependent on, like pyroptotic molecules. It's not necroptosis because it's, of course, not driven by this. And we asked, what is the role for this complex? And it's really fascinating that the same TK1 knockout that provided protection in bone disease, in skin disease, is also providing protection from cell death. So what is this telling us again? That caspase-1, which is inflammasome, is redundantly working with the caspase 8 and RIPK3 in driving cell death. Right. So overall, this study discovered the first time that ZBP1 is innate immune sensor that is sensing nucleic acids or the damage that's done by influenza virus. And once ZBP1 is upregulated, it forms a multi-protein complex where inflammasomes are integral to this complex for driving IL-1 beta IL-18. But the cell death is not pyroptosis or it's not necroptosis. It's not obviously apoptosis because this is non-lytic pathway. But the death pathway induced by ZBP1 is fully lytic, and we thought of thinking about putting together a conceptual framework for all that we have been seeing for a decade. And because the molecules that are involved in death are pyroptotic molecules, apoptotic molecules, and also RIPK3, RIPK1, so we began thinking about calling it or this pathway as panoptosis. But we were extremely careful, and it took us another 5 years to really define this pathway. But ZBP1 laid a foundation for a new research frontier in innate immune cell death regulation. So overall, our study provided the first evidence that ZBP1 is actually could act as an innate immune sensor of influenza virus triggered NLRP3 inflammasome activation. And we thought at that time it's activating multiple program death pathways, but we know now that it is activating panoptosis. So overall, this study provided the genetic evidence for the role of ZBP1 as an innate immune sensor, inflammasome regulator, and it drives the complex activation of caspase 8 RIPK3 complex. And it didn't happen overnight, like, for the discovery of ZBP1 happened over a decade because it began with NLRP3. We discovered influenza virus as a trigger for inflammasome activation. And we started asking what is upstream of influenza virus, and that's when we discovered ZBP1. So it took us almost a decade, 10 full years to really discover ZBP1 as a sensor and cell death regulator. And then we began asking, does it happen in single cells, right? So we can see here that upon influenza infection, the pyroptotic component like ASC is co-localizing with that of caspase-8, the apoptotic molecule, and with that of RIPK3, the necroptotic molecule. And the PAN complex, the panoptosome complex, is forming upon triggering with influenza infection. And then we don't believe in one line of evidence. We need multiple lines of evidence. And we also looked for another model like expansion microscopy, the new technology at that time that came. And even in expansion microscopy, in single cells, we're able to show the co-localization of caspase-8 together with ASC or caspase-1 and together with RIPK3, suggesting that also, laid the foundation for the panoptosome concepts. So overall, as we discussed, the lytic pathways are so far pyroptosis, which is inflammatory caspases dependent, Gas-Domin dependent. And another lytic is necroptosis that happens only when caspase-8 is absent and the executioner is MLKL. And then we defined another lytic, new innate immune lytic pathway, which is panoptosis. So what is panoptosis? It is an innate immune inflammatory cell death pathway where an innate immune sensor like ZBP1 senses PAMPs or DAMPs or pathogens and nucleates a complex together with caspase-8. So caspase-8 is a key molecule that brings all the components like caspase-1, ASC, or RIPK3 into the complex. And this is very unique because in contrast to other pathways, here we can see caspases activation. We can see inflammasome plays integral role for IL-1 beta maturation in these complexes. RIPK1 is involved and RIPK3 is involved. So we defined this as a pathway initiated by sensors, as we discussed, driven by caspases and RIPKs. Even though we know caspase-8 is a major one, in the future we do not know because not much is known about other caspases and their roles in lytic cell death. So we kept the definition very broad open because depending on the trigger, it could be other caspases that might be involved. So we let it as caspases and RIPK complex, which is panoptosome complex. So in the beginning, it was really tough. It's against the dogma. We were breaking all the rules before. And then after our publications, there were many, many studies published following up on our studies on ZBP1 in Nature, in major journal, Cell Science, in many journals. And then also, when we first showed that there is role for caspase-1, none of the folks in the field believed it. That's why our manuscript ended up in 2014 in JI. But then you can see after several years, in 2019, several Nature papers appeared exactly suggesting the same thing, that caspase-8 can nucleate these molecules. So you can appreciate how dogmatic the field is because initially what we know is non-lytic apoptotic pathway and there is lytic, like necrosis, non-programmed death pathway. So for pyroptosis, for Brad Cookson and Brennan to say that there is another programmed lytic cell death pathway, it took them I do not know, for 5 results to appear in PubMed. It took them 6 years to get to 5 results. That means people didn't believe in this pathway. But we're very happy that panoptosis is filling a major gap in the field, that in 6 years, within 6 years, there are close to 600 results if you type in PubMed for panoptosis. So then ZBP1 is a founding member, where upon infectious agents, then ZBP1 forms a multi-protein complex called panoptosome, where multiple executioners can be cleaved, driving panoptosis, which is causing membrane damage, or several pore-forming molecules might be also cleaved, releasing a lot of DAMPs, cytokines, inflammation, and leading to disease pathology. So after this, then we began thinking, how do we search for other panoptosomes? How can we discover new sensor molecules that can engage these multi-protein complexes that drive this lytic cell death? So that led us to think about what could be the possible combinations of or physiologically relevant conditions where cell death could be triggered. So during COVID, we asked a very simple question about, are there any cytokines that are triggered downstream of innate immune responses? Because you all know TLRs can release TNF, can lead to TNF production, right? And then gamma can be produced by NK cells. So we were trying to mimic in vivo condition to see whether there are any cytokines that can induce lytic cell death pathway. And as you can see here, low doses of TNF or interferon gamma do not induce cell death, but the same low doses of TNF and gamma, when combined, they induce robust cell death. And it happens both in human cells and in mouse cells. So this combination, only this combination was induced cell death. So we asked, what is the nature of that cell death? And obviously, it's not pyroptosis because none of the pyroptotic molecules are involved. It's not apoptosis because it's lytic, and none of these caspases were involved. And then it's not necroptosis because none of the molecules involved in necroptotic pathway, MLK, LRIPK3, are involved. And again, the same 3KO that was providing protection in multiple diseases, the node caspase 8, caspase and RIPK3 deficiency provided protection. And then during that time, we were able to show that there is synergistic effect of downstream cytokines regulated during innate immune responses could also drive this inflammatory feed-forward loop, like cytokine storm, instigating multi-organ failure and death. So overall, we were able to show that STAT1, IRF1, nitric oxide are upstream of this pathway in assembling panoptosome complex and in driving panoptosis. And this is really critical to identify the pathway molecules so we could find inhibitors. And in those days, actually, baricitinib, based on our study, I think received emergency approval to treat COVID. And this blocks because JAK inhibitor, STAT inhibitors are upstream of this panoptosome pathway. So how do we find other triggers? We started after our studies opened up a new way of thinking for us, that in physiologically relevant conditions, there will be PAMPs and DAMPs combination. So we showed that single PAMP or single DAMPs do not induce cell death. But the combination, like if we combine heme that is released in hemolytic conditions together with LPS, PAM3, or TNF that is induced downstream of LPS sensor TLR4, can lead to robust cell death. So this gave us an indication that there could be a sensor upstream of sensing this combination, which is physiologically relevant. And then we screened maybe 200 different knockout strains that we have of all the sensor molecules, be it TLR, C-type lectins, or C-gasting pathway, RIGI, NLRs. And after screening many molecules, asking what is the sensor and what is the death pathway, we discovered that NLRC5 and NLRP12 are involved in this. So these are the same molecules I started off as a postdoc. At that time, I couldn't really discover any functions for this because we were believing in the dogma that only one trigger can engage one sensor. We never really appreciated the concept of network of innate immune sensors driving this disease upon sensing PAM-DAM combinations. So here we also showed the TNF downstream of LPS TLR4 signaling also can induce robust cell death. And then interestingly, in hemolytic diseases, we also have seen NLRC5 P12 being highly upregulated. And then what is the nature of cell death? So now we are asking whether the panoptosome complex is involved and if the death is panoptosis. So how do we test that? We test by asking the role of caspases and RIPKase. So as you can see here, heme together with PAM3 can activate caspase 1, inflammatory pyroptotic molecules like gasoline D, E, and also this combination activates caspase 8, caspase 3, caspase 7, and also MLKL. This is a signature of panoptosis. And then we show that in the absence of NLRP12, there's absolutely no caspase 1, gasoline D, E, caspase 8, 3, 7, MLKL phosphorylation. So this is the key signature to suggest that this death is panoptosis. And we showed NLRP12 and NLRC4 are acting as like sensor molecules sensing this PAMP-DAMP combination to induce this panoptotic pathway. And we showed in vivo that if we delete NLRC5, there is less inflammatory cell death panoptosis, reduced inflammation, and mice were rescued from lethality. And we also asked what is upstream of NLRC5 and P12, and we were able to show that NAD depletion and ROS could act upstream of the sensor pathways. This again brings the new concepts of a metabolite alteration in the cytosol could be also sensed by these NLRs or cytosolic innate immune sensors. And if you can appreciate upon this, like mimicking conditions, physiological relevant conditions like heme or PAMs or cytokines combination could begin sensed by TLRs, TLR4, TLR2, and the cytosolic sensors. There's a beautiful network of innate immune sensors assembling the complex, again, driving the network of executioners, driving pore formation or membrane damage. So this is the concept that's there in the plant field, that NLRs form a network. And here we were able to show that heme plus TNF can activate this panoptosome complex. So overall, we began with ZBP1, which is sensing IAV infections. And we also showed that AIM2 can act as inflammasome if you transfect DNA, but if you go with full pathogens mimicking in vivo conditions, then AIM2 assembles panoptosome complex. And here in Yersinia infection, our TACI inhibition, we showed that RIPK1 acts as a sensor, innate immune sensor, forming a protein complex. And then, like a downstream executioner network is also cleaved, because you can imagine caspases in the complex can go on and cleave multiple proteases or phosphorylated molecules downstream, damaging the membrane and then leading to disease pathology. So then we went back and asked very simple question. How about NLRP3? Historically, I began my career studying NLRP3 as an inflammasome, right? That's what we discovered. But then with this open concept of PAMP, LPS, and a DAMP combination, we began asking, is it really an inflammasome? So, or is it a panoptosome, right? So it's a very simple question we're asking, and NLRP3 and ASK deletion completely provided protection. One would think it's inflammasome, but then, as you can see here, to our surprise, as soon as after 30 minutes, caspase-1 deficient cells start dying. Gasdermin D, gasdermin E, even caspase-8 TRPK3 cells, deficient cells start dying, and the same TKO complex provides protection. And we have to actually ask the question, what is happening in wild type cells? So as you can see here, in wild type cells, as soon as 15 minutes, you can see beautiful colocalization of NLRP3, the caspase-8 and RIPK3, our core complex of panoptosome, are coming together, forming a panoptosome complex. And this complex is happening even in the absence of caspase-8 and gasdermin. So people talk about, misleading things as, if you block caspase-1, it goes to other death pathways. Like, what are the conditions where actually caspase-1 is deleted in human beings? It's not. So we have to go back to the wild type setting and ask, what are the molecules that are activated in wild type setting, and ask this question, what is the complex, the combination that's providing full protection? And that's what the importance of the complex. So now we know that NLRP3 panoptosome, within this panoptosome, inflammasome is activated for IL-1 beta maturation, but the complex is formed to drive lytic cell death that is independent of pyroptotic molecules like caspase-1, caspase-11, or gasdermin D. So there's network of sensors here and network of executioners. And then it's really amazing to see that one IRF, which is IRF1, is acting upstream of this assembly of complexes, transcriptional translational regulation of complexes. So far we have discovered 5 different panoptosomes like ZBP1, AIM2, RIPK1, NLRC5, and P12, and also the newly added NLRP3 panoptosome. And this reflects what we see beneath the surface. And, this is my last slide, where we always believed that a single ligand receptor model is what we believed in. But this is oversimplification of true physiology, because you can see during infections, lots of PAMs are released, lots of DAMs are released, cytokines are produced, tissue damage is happening, cell death is happening. This is what is happening in real world. That means there is network of these sensors beginning from TLRs to TLRs producing TNF or other interferons, and these molecules transcriptionally, translationally regulating these innate immune sensors, forming this network. And this network is essential for assembling a panoptosome complex that consists of caspase 8, caspase 1, and then once caspase 8 RIPK3 caspase 1 complex is formed, then these pore-forming molecules are all cleaved simultaneously. There's no need for, cleaving only one molecule at one time, because the cell has decided to die in a lytic way so that it can alert the immune system. And also downstream of this lytic cell death pathway, then more and more PAM stamps are available for engaging more sensors, amplifying this. So this single ligand receptor model is complete oversimplification of true physiology. So that's where the concept of paroptosis, which is innate immune inflammatory cell death pathway that is driven by caspases and ripcase through panoptosomes, make complete sense. And these are the multi-protein complexes assembled by innate immune sensors. And then this captures the multifaceted nature of innate immunity, as you can see here, multiple PAMs and DAMs that coexist that we always ignored. And then this panoptosis is initiated by a network of innate immune sensors, which is very, very well validated in plants. In plants, there's a network of sensors are formed. And this assembles the panoptosome complex that activates again a network of cell death executioners. So who says it's only gastromin D or E or MLKL phosphorylation? We believe that there are other pore-forming molecules yet to be discovered, or membrane-damaging molecules yet to be discovered, that can be cleaved, instigating this inflammatory disease. And we have to remember that apoptosis is non-lytic, non-inflammatory cell death initiated by initiators executioners. And pyroptosis, to respect Dr. Yerchop and the original definition, it is mediated by inflammatory caspases, which is caspase 1 or 11 dependent death. And if the cell death is independent of caspase 1 or 11 or inflammatory caspases, then it is not pyroptosis. And none of these pathways are necroptosis, because caspase 8 is central to this. It's driving this lytic cell death pathway. And this is totally independent of MLKL or gasdermin, so it's not necroptosis or it's not pyroptosis. So this fills in a major, major gap in the field, and we'll believe this is the reason why we couldn't really find therapeutic targets. But now we know how to really visualize physiologically relevant conditions, which we began saying that there are limitless number of triggers that we get exposed to, and how we sense via network, how complexes are assembled depending on the sensor molecule, and how death is executed, and where multiple gasdermins or pore-forming molecules, membrane-damaging molecules are involved in instigating in immune responses. So with that, I would like to thank all my former members and current lab members for their contribution, because this was a very, very tough work. We were going against the dogma, challenging all the rules. So I'm happy to take any questions. I'm sorry if I have gone over the time. And I'll stop there.
So thank you very much, Dr. Kanneganti. It was an amazing talk. So we always give opportunity for the students to make the first questions here. So do we have any questions here? São Paulo, Rio, Uberlândia? Yeah. So Gustavo can open up the questions here, right?
Hi, Thiru.
Hello.
Wonderful presentation. I miss you so much, really, your
Me too.
insights and this beautiful work. I have so many questions, but of course I will select and maybe start with the more general. And I think the first question is regarding your last slides. So do you envision any situation where pyroptosis or necroptosis would physiologically happen in the relevant context? Because apparently, all pathogens are very complex, and if they trigger, panoptosis. So how do you feel about this?
Yeah, that's a great
And just
question.
adding another flavor, are all these circuits happening in the same way in all cell types, or let's say macrophages are, more prompt to get this circuitry on?
So these are all great questions. So first one is, what is the role, physiologically relevant role for inflammasome or necroptosis? So inflammasomes, to me, their main role is for maturation of IL-1 beta, IL-18. So that is to alert cells before even cell death is induced. So cell death is a compromise for our host to really let our own cells die. So to me, IL-1 beta and IL-18, these cytokines are alarm, danger bells to let our system know that there is danger. And also under certain circumstances, when maybe there is a pathogen or a situation, maybe mutations, right? So for example, NLRP3 mutations, there it makes sense completely that IL-1 beta is the cause for the disease. So inflammasome plays a role. So I believe that there are those unique conditions where there are patients with mutations in NLRP3, for example. And then for necroptosis, there could be some pathogens under rare circumstances could carry inhibitors of caspase 8. That's how necroptosis was discovered by Dr. Ed Mokaczki and et al. Right? So there are conditions where when pathogens carry inhibitors of caspase 8, that's when maybe MLKL drives this necroptotic pathway as a backup mechanism. But the most prominent mechanism to me is this innate immune cell death, which is panoptosis for driving cell lysis. So I think that distinguishes necroptosis with that of panoptosis, and also inflammasome role for primarily IL-1 beta, IL-18 maturation. Then the next very important question is, what are the cell types that undergo these various types of cell deaths? So of course, innate immunity is mostly predominantly run by immune cells like macrophages, dendritic cells, like neutrophils. But then I'm visualizing, let's say, for example, a pathogen is inhibiting caspase 8, driving necroptosis. That could also provide more DAMPs, right? That could also instigate panoptosis one way. The other way is actually depending on the molecules that are expressed in different cell types. Cells might be undergoing cell death in a different way. So most immune cells are equipped with innate immune sensors. Most immune cells are equipped with caspases. So they drive predominantly by panoptotic pathway. And epithelial cells, maybe they die in a predominant way with necroptosis, maybe, because they might not express NLRs or they might not express inflammasomes or caspase, inflammatory caspases. So this multiprotein complex formation depends on whether sensors are there in those cell types. But then even when necroptosis is instigated or other lytic pathways are instigated, I believe they again engage panoptosis, because more PAMs are released, more DAMs are released, more cytokines are released. So this is also a feed-forward loop that could instigate at the end panoptosis. So long answer, but I hope I clarified.
Oh, it was wonderful. So I have other questions, but I will allow other people to ask, and then I may come back. Okay, thank you so much.
Thank you.
So Bruno raised his hand here, and we have some questions on chat as well.
Yeah.
Do you want to go
Wanna
ahead,
go ahead?
Bruno? Or should I go to the chat?
Oh,
I
I
was
can.
I was the
Yep.
one that is in the chat, or is it from a student there, Bruno?
No, actually, no.
Okay, so
It's from me.
I was the one that's in the chat from students. So Ruth wrote, wonderful presentation. I'd love to ask, is it possible for ZBP1 to induce IL-1 beta release through a caspase-1 dependent mechanism that is independent of canonical inflammasome components like NLRP3 and ESC? If so, what are the proposed intermediate platforms or adapter proteins that might facilitate this non-canonical activation of caspase-1?
Yeah, so that's a great question. So ZBP1, once activated, it engages inflammasomes, right? So inflammasomes, depending on the trigger, they are integral components of panoptosomes. So IL-1 beta is released downstream of panoptosomes via inflammasomes. So that's the clarity that we have been providing through our data. And then, as we discussed, in certain patients, we don't need ZBP1. It's NLRP3 mutations can actually lead to overactivation of caspase-1. And then your other question is the nucleation of small complexes. So I couldn't believe in our data that within 5 to 10 minutes, within 5 minutes maybe actually, we can see caspase-8 TRPK3 co-localizing with caspase-1. Right, or ASK. So what is this telling us? This is really telling us that maybe caspase-8 TRPK3, caspase-1 ASK, they're all nucleated in cell in a time-dependent manner, and they're all coming together. So it would be really an amazing open research area to understand the biology of these complexes, when they are formed, how are they formed, and you will get there only when we use physiologically relevant conditions.
Great. So Bruno, you want to ask there?
All right. Thank you very much for your talk. It was really a tour de force, 200 mouse strains. It's no joke. It's a lot of work, but here I come to ask for something else that you may or may not have done. So from systems biology and physiology perspective, you would also expect to have some sort of feedback loop in the system to dampen its response and allow the system to go back to homeostasis. Is this feedback loop in panoptosis is something specific? It's something that is generic response due to tissue damage. Do you have any insight on that part of the equation?
That's a very good question, and that's what we all must be thinking, right? So we have to go back and really check to understand in tissue damage conditions versus infectious conditions. So not all pathogens are the same, and not all pathogens induce robust cell death. So it depends on conditions where we see lytic cell death. That's when we have to ask what type of cell death is that. So I think when cell decides to really undergo cell death, that is a decision that's made thinking that it's good, but depending on the pathogen, depending on the force of cell death that happens could be really detrimental to host. So as a simple example, I can give COVID, right? So under certain conditions, people recover and it's fine. But under certain conditions, COVID led to many deaths also, right? So this is a feed-forward loop or amplification of cell death that's leading to this severe inflammation. So in septic shock conditions, in hemolytic conditions, in severe pathogenic conditions or underlying conditions, it could lead to panoptosis and serious inflammatory conditions. But then are there any feedback or inhibitors of this pathway? I do believe there might be inhibitors of this pathway that we have to discover. So we are not there yet. So we all have to search for these inhibitors or search for conditions where panoptosis is induced. It's a completely wide open field. So there's a lot to discover and a lot to figure out.
Okay.
Right. Thank you very much. I think we have questions here on the chat.
We have two questions in the chat, right?
2 questions actually. Should I go ahead, Diego?
You can go ahead for the first one, yeah.
Sure. So the first one is from Barbara. What's your take on panoptosis and tumor resistance to treatments? Do you think tumor cells have natural inhibitor pathways of panoptosis?
Yeah, so I think that's a very good question, a great question. The best way to kill tumor cells would definitely be panoptosis, so that we can make sure the cells are dead, because TNF interferon gamma combination, we showed in one of our manuscripts that we looked at NCI 60 different cancer cell lines. Most cancer cell lines can be killed by using TNF and interferon gamma combination. So if in the tumor microenvironment, it would be great to let these cells die in a panaptotic way. So we really do not know exactly under what conditions or in what cancer types that panaptosis can be targeted is not known, and we have to really understand. And also when people use cell lines, that lead to a lot of artifacts, right? A normal cell has become a cell line. That means it have the cells have kicked out the most critical genes in cell death pathway. Otherwise, it would not have become a cancerous cell line. So if you try to use cell lines to understand the biology, it would be very difficult. So we have to make sure that our components are there. In some cell lines, ASC is not even there. In some cell lines, RIPK3 is not even there. So always we have to first test what are the molecules that are expressed in a particular cell line or a cell type, and then we can study the pathway.
Right. Thank you. And we have also another question from Clarissa Maia Monteiro. Thank you for the wonderful work. I would like to add something from the beginning of the work. I may have jumped. I may have missed it, but I'm curious to know how the caspase activity is activating the NALP3 receptor. We always think of the other way around.
NLRP3 is activated. It depends on what it senses, right? Usually the consensus is any membrane damage leading to NLRP3 activation. So what we think is, depending on a pathogen, depending on a receptor that's sensing the pathogen, then it can assemble NLRP3 as an integral component for driving IL-1 beta maturation. So this is, genetic evidence is what is telling us. And I'm with you all to keep it simple, right? Keeping it simple is always easy, but then it's not real biology, it's not real research, it's not real discovery of anything. So, you have seen how complex it is. So we cannot keep things simple, because in vivo, nothing is simple. It's all a network of molecules that get upregulated. If you look at your RNA-seq, there are you can see hundreds of genes that are upregulated. But then we have to nail down to the molecules that are involved. So I didn't address your question right away. Stay in a straightforward way. But depending on the trigger, they could act as a sensor molecule, or they themselves could act as a receptor as well.
All right. Thank you. Diego, back to you.
Yeah, so do we have more questions? Actually, Clarissa actually made a comment. I still restrained on the simple idea from the receptor to the enzyme, right? As a complement to the previous question. So do we have extra questions here? Gustavo has another question.
Hello again, Thiru. There are a couple of things that I'd like you to clarify to me. One is there any other requirement in the cell undergoing panoptosis to avoid apoptosis? So is there any need for, let's say, relatively high levels of AIPs or something else? Because as I understand, there's no sign of this is an inflammatory cell death, so there's no really apoptosis going on. And together with this, are caspase 3, 7 activated during panoptosis?
Yeah, so that's a great question, and we really need to check the involvement of other apoptotic molecules in lytic cell death pathway. So we went with the dogma for decades, not touching all these caspases for any of their roles in lysis, right? So in our complexes, like caspase 8 is central to this complex. So we see beautiful activation of caspase 8, and as soon as caspase 8 is activated, caspase 3 is activated, caspase 7 is activated. So we could see 3, 7, 8 all are activated in our pathway. So what is the role for other molecules that are very well established for apoptosis? We have to revisit all of them to see whether they have any role in driving lytic cell death pathway, which is a programmed pathway downstream of innate immune sensing.
Right, so caspase 3 and 7 are activated, but you don't see the signs of apoptosis like blabbing or things going on. This is, do you believe this because of dynamics or just the speed of things happening, or you believe there's a blocker? And is there any role for BCL2 mitochondrial regulation?
Mm.
in this
So
regard as well?
yeah, so BCL2 mitochondrial regulation is open question. We're really interested in checking that possibility. And then in a single cell, when the sensors are engaging caspase 8, caspase 1 complex, it will immediately start cleaving the substrates that drive towards lytic pathway. So that's why I believe that as soon as caspase 8, caspase 1 are involved, gasded means left and right are cleaved, so that leads to membrane damage and membrane pores. So in the process, you're asking a very interesting question. Do you see blebbing? Always before lytic cell death, we see the bulge of cell, right? And then it will pop. So the dynamics would be really interesting to check what happens. But then I do believe that as soon as caspase 8 and caspase 1 are cutting cleaving lot of these pore forming molecules, membrane damaging molecules, the cell doesn't need to die in an apoptotic way.
And challenging for you. Yeah, it was very surprising in a way of the role of combination of interferon gamma and TNF alpha.
Yes,
Your
yes.
data is very, very clear,
Mhm.
but these are two cytokines that are produced systemically, quite a lot during infections.
Mhm.
So what would be why you wouldn't damage just in an extreme model such as the SARS-CoV-2, but not others?
Yeah. So the simplest explanation for that is, of course, TNF is NF-kappa B mediated, TLR receptor mediated. But then most immune cells produce TNF, but not gamma. So macrophages do not produce gamma. So we believe that only under certain circumstances or in certain microenvironments that gamma is produced to drive maybe T cell responses. So this compartmentalization always keeps these two cytokines separate. Right, Gustavo? So that's what we are thinking. And that's why the combination doesn't happen unless and until in a rare conditions where cells are really, dying, TNF is there, and then NK cells are producing gamma. That combination becomes detrimental now.
Like in tumor microenvironment.
Mm-hmm.
So,
Yeah,
okay.
so it's very interesting to really understand the compartmentalization and then under what conditions these two, come together. Because none of the immune cells produce interferon gamma. Most immune cells, macrophages do not produce gamma.
Okay.
Yeah.
Many thanks, Thiru, and hope to see you very soon.
Yeah, thank you. Many, many things to discover. So I hope, we all work towards that understanding. Thank you all.
Bruno, do you have a final question there,
Yes,
or?
please go ahead. Yeah, yes, I would
I'm
love
sorry.
to.
We actually have one more question from the chat from Marcelo Basinski. He says that it was an impressive lecture. And if you could please elaborate on the selective advantages of adaptive immune response.
So we really didn't get that far to understand innate immune sensing molecules or caspases in adaptive immune responses. That's again a major gap in the field between innate immunity, cell death, and adaptive immune responses. So that's a wide open research area. So I cannot comment because I never looked at exactly the T cells or adaptive immune responses downstream of innate immune responses.
All right.
And of course,
Thank
we
you very
always
much.
believe that innate comes first, and then that instigates adaptive. So, and then cell death isn't really in the middle.
Thank you.
I think we're done with questions here. Would like to thank you very much again, Dr. Kanneganti. It was an impressive lecture. I think everybody here loved it. It was an enormous amount of work and data and all the history on inflammasome activation and cell death modulation. So very good. Thank you very much.
Thank you so much. Please feel free to send emails if you have any questions or if I can clarify anything further. I would love to hear from you. Thank you all. And see you,
All right.
Gustavo, and say hi to Karina and all the rest of the friends in Brazil.
All right. Bye-bye.
Thank you.
Thank you.